So, what we know about cervical cancer? First, it is well known that HPV causes cervical cancer in humans, HPV being human papillomavirus. Less known is that disease develops only in small proportion of infected population, and it may take a long time, years and sometimes decades.
In fact, this virus causes a chronic infection as a result of a weak antiviral response generated by women and immune inhibitory properties of high-risk HPVs. The virus has two genes, E6 and E7, that cause genomic instability and chromosomal aberrations. Simultaneously, HPV integrates into the human genome, although it is still present in the episomal form, with E2 keeping low the expression of the integrated E6 and E7 oncogenes.
Paradoxically, despite a weak immune response, episomal virus is gradually declined and the integrated form is increased during the disease progression.
So why would a woman who was unable to eliminate episomal virus for a long time, sometimes decades, now acquires the ability to abolish HPV in the tumor?
It turns out the tumor itself orchestrates the antiviral immune response.
But how does it do this?
In the process of tumor evolution, chromosomal aberrations provide a genetic source of adaptation. This applies to cervical cancer as well as to any other cancer. Indeed, recent meta-analysis demonstrated several regions of consistent gains and losses in genomes of cervical tumors. Consistently with the evolutionary process, the most frequent aberrations are found early during development of precancerous lesions.
Why do these aberrations appear at specific places in the genome?
While genomic instability might affect random loci, only those cells that harbor aberrations that give advantage for tumor growth would be further selected. Such survival advantage is provided by the drivers of cell cycle and antiviral response located in these chromosomal gains.
To put this all together, based on our and other group results, we proposed a model in which selected chromosomal gains simultaneously contain key gene drivers of antiviral response, cell cycle, and microRNA inhibitors of epithelial differentiation. On one hand, cell cycle drivers sustain continuous cell proliferation and growth. On the other hand, antiviral drivers overexpressed in the tumor trigger an immune response which reduces the expression of the episomal virus and E2 activity in the tumors. This would release E6 and E7 oncogene expression, leading to the block of p53 and retinoblastoma. Thus, these three processes ultimately synergize, promoting uncontrolled tumor growth.
This model, as well as some recent data needed for its generation, is a result of a large collaborative effort between the teams from Norway, Brazil, and the United States.